Okay, so today we are going to see sexual reproduction in flowering plants. Okay, so what is sexual reproduction? Sexual reproduction involves the union of gametes. Okay, so there are two gametes, the male gamete and the female gamete. So the male gamete is the sperm, the female gamete is the egg. So the fusion of these gametes is known as fertilization, which will lead to the sexual reproduction. Okay, so the basic trivia is that the egg will be the largest cell in the human body, while the sperm will be the smallest one. Here, okay. So the sperm will be mobile, but it will be small. So sperm is the gamete which can move. So it is mobile and it is small, whereas the egg is immobile since it is large. Okay. So during fertilization, what will happen? They will unite. The male and the female gamete, they unite. So each of them will have n number of cells which will form a diploid. Diploid means two times that of the n. That is two times. Diploid means two. So two times that of those cells will be formed, which is nothing but a zygote, the fertilized egg. So this is the basis of the zygote will then form the embryo and which will give rise to a young one. So this is the basis of sexual reproduction. So now we'll see about sexual reproduction in flowering plants. Okay, so what are flowering plants? Plants which are capable of producing flowers. They are called as flowering plants. So this flowering plants, they comprise 90% of the plant species. Okay, so what are flowering plants called as? They are called as angiosperms. Clear? So flower is a fascinating organ of the angiosperms, and we had an intimate relationship with flowers. So it has been known as an object of aesthetic, ornamental, social, for religious use, for cultural values. So for everything, we convey our human feelings using flowers, like even love, affection, even for grief, mourning, everything we use flowers. But do we know that flowers are actually the aid of sexual reproduction of the aid of sexual reproduction? So all the flowering plants, they show sexual reproduction. And look at the diversity of the structure of the inflorescence, the flower, the floral parts. So it will have a range of adaptation to ensure the end product of sexual reproduction, that is the fruit or the seed. So finally, what is the end product of the sexual reproduction? It is nothing but the fruit and the seeds. Clear? So 90% of the plant species is nothing but flowering plants. All of them we call it as angiosperms. Flowers are the aid of sexual reproduction. Flowers are produced as a result of sexual reproduction. All the flowering plants, they show sexual reproduction. And the final end product of the sexual reproduction is nothing but the fruit and the seeds. Okay, so now we'll see the concept of this lesson, sexual reproduction in flowering plants. So there are three stages in sexual reproduction in flowering plants. First is pre-fertilization, then we have the fertilization stage, then the post-fertilization stage. Clear? So what fertilization is the important stage here. So fertilization means the fusion of male and the female gamete to give rise to the young one. So this is the main stage here, and this the stages which occurs before fertilization is pre-fertilization. Before, so pre-fertilization, and which occurs after fertilization is known as post them. First is pre-fertilization. Yeah. Okay. So what is the reproductive organ in plants? Flowers. Obviously, the flowers are the reproductive organs or the structures of a plant. So most flowers, they are hermaphrodite. So what does this term mean? Hermaphrodite means they have both male as well as female sex organs. So single flower, they'll have both male as well as the female sex organ. So they are hermaphrodites. So most of them are hermaphrodites. Now, what are the parts of a flower? Okay, so the two main parts of the flower is the stamen and the pistil. Okay, so the stamen is the male reproductive structure. It comprises of the anther and the filament. Okay, and the female reproductive structure is your pistil. Okay, this comprises of stigma, style, and the ovary. So this is the stigma, this is the style, and this is the ovary. Okay, so these are the main parts, the male reproductive structures and the female reproductive structures. Okay, so apart from these, there are accessory organs. Okay, they are the corolla, calyx, receptacles, and peduncles. Okay, so accessory parts. So what is the function of these accessory parts? The main function of these accessory parts is giving support for protection as well as to attract during pollination and all that the bees or any insects must be attracted. So attraction. So these are the functions of these accessory parts. So it is for support, protection and attraction. Clear? So we'll see each one of them one by one. First is your calyx. So what is the calyx comprises of? It comprises of the sepals. Sepals, here you can see the green color structures. They are called as the sepals. They are leaf-like structures. Okay, and the main function is protecting the flower when it is young. You can see a flower bud. How will it be? It will be covered with the calyx. Okay, so for protecting the flower when it is young, when it is still like a bud, this calyx helps in protection. Clear? Next is your corolla. So corolla comprises of the colorful petals. So corolla is nothing but your petals. So these petals, they can be of various shapes, size and colors. So and the main function is attraction and protection to the inner reproductive structures. Okay. Next is your receptacles. So this part is the receptacle. It is also known as torus. Okay, and the main function is supporting the entire flower. You can see it is a the base of the flower. Base of the flower எப்படி இருக்கு? It is like a பெருசா like அந்த மேல இருக்கிற part support பண்ற அளவுக்கு it is heavy. Okay, so it is swollen and heavy at the base. So this receptacle is only helping the entire flower, to supporting the entire flower. Clear? Next is the peduncle. Okay, peduncle is also known as the stalk. So this part is the peduncle. Okay, so main function of peduncle is it connects the flower to the branch or the stem. So this peduncle will connect the flower to the branch or the stem. That is the main function of peduncle. Okay, so till here we have seen the accessory parts, the calyx, corolla, receptacles and peduncles. Now we'll see the reproductive parts. Okay, what are the two main reproductive parts? They are the stamens and the pistils. Okay, so the male reproductive part is your stamen and the female reproductive part is your pistil. So this is the stamen here and this is the pistil here. Okay, we'll see each one of them. First we'll see about the stamens. Okay, so the male reproductive organ is the stamen. They are collectively called as andrecium. Okay, so the male reproductive organ is nothing but your andrecium. We call it as andrecium. So what are it composed of? It's composed of two main parts, the anther and the filament. Okay, so this is the anther and this is the filament. Okay, now what is the anther? It is an expanded lobular structure at the end of the filament. So this is the filament here. So at the end of the filament, there is an expanded lobular structure. So what is lobular? Lobular means a circular or flattened projection which hangs outside. Okay, so it is an extension of the filament which is hanging outside. That is the anther. Okay, expanded lobular structure at the end of the filament. So what is its function? Its main function function is to produce the pollens, which are the male sex cells. Okay, so function is produced. What is a filament? It is a stalk that supports the anther. Okay, the stalk which supports and holds the anther is called as filament. Clear? Okay. Now we'll see the structure of the anther in detail. Okay, so this is the cross section. When we cut the structure of the anther will be like this. Okay, so how will it be? It will be bilobed. So it has two lobes. So it is known as bilobed. Bi means two. Bilobed. Other rendu lobe. Okay. Then it is dithecus. So it has di means two. So dithecus. Now what? It has two thecas. Thecas means cell or compartments. It has two cells or two compartments. Okay, so it is dithecus. Okay, you can see it is bilobe. It is dithecus. Okay, and it has a longitudinal groove. Groove na enna? It is a depression or a cavity or a depression is called as groove. ஒரு ஹோல் மாதிரி இங்க உள்ள வந்து ஒரு ஹோல் மாதிரி ஒரு ஸ்ட்ரக்சர் இருக்கிறது தான் வந்து groove. Okay, so it is we call it as four-sided, so it's tetragonal. Okay, and it has four microsporangia. Okay, so microsporangias are nothing but the pollen sacs. So we have four pollen sacs, four microsporangias. Okay, and inside this microsporangia the pollen grains will be formed. Clear? So this is the structure of the anther. So it is bilobe, dithecus. It has a longitudinal groove. It's four microsporangia in which my pollen grains are formed. Clear? Okay. Now we see the structure of the anther wall layers. இப்போ வந்து ஆந்தரோட அந்த 3 அந்த வால்ல we'll see what is the structure of the wall and the wall layers. Okay, so outermost and inside then we have the middle layers. They have atom, then finally have genus tissues. Okay, where the microspore mother cells are present. So apitum, then porogenous tissues. Okay, so this is okay. Is my voice breaking now? Now also you can't hear? Okay, you can hear. Now can you hear me? Okay, very sorry. Here it is heavily. Okay, I'm starting again. Okay, from where you people, I mean, where did the connection cut? From where shall I start? Anyone you can unmute and say from answer wall. Okay, okay. Okay, I'll start from answer wall. Okay. Okay. So the answer, so the outermost layer, which is the epidermis, then capita, and finally the clear. So these are the different layers in the anther wall. Clear? Okay. Now here, first the outermost layer, the epidermis. The outermost, the main function of epidermis, dehiscence of anther. Okay, so what is dehiscence? So it means splitting. So you have splitting here, okay, in the anther layer, anther, which is giving a what to say, a four-sided structure. That is kind of dehiscence. Okay, that is called as dehiscence. So chromium will be present. Chromium is structure which is present between two fossats and present at the chromium region. The endothecium. Okay, so endothecium. Okay, below the epidermis is the endothecium, and this endothecium is also one cell thick. Okay, it is one cell thick. What is it composed of? It is composed of fibrous cellulose thickenings plus a little of pectin. Okay, cellulose and pectin. Okay, the main function is at maturity. What will happen? These cells, these endothecium cells, the water present in it. Okay, and they will help in the dehiscence of anther. The cells will shrink, and there is a different, is creating a crease or the splitting of the anther, the dehiscence of the anther. Clear? So we saw about epidermis. This is endothecium. Next is the middle layer. Okay, so below the endothecium is the middle layer. So here it is the middle layers here. Okay, so the function is at maturity, what will happen? These cells will degenerate, and it will help in the dehiscence of the anther. Same function, and along with that, it will also help the protection of the anther. Okay, dehiscence and protection of anther. Next is your tapetum is the innermost layer. This is considered as the innermost layer, but still you'll have the sporogenous tissue surrounded. Okay, here the cells are multinucleated. Okay, there'll be multiple nucleus and it is polyploid. It contains some structures called as Ubish bodies. These Ubish bodies will secrete sporopollenin. Okay, sporopollenin. We'll see what this sporopollenin in the capital. So the main function is meant to the developing pollen grain. Okay, so this is you wish developing pollen grains. Okay. So microsporogenesis. Okay, so as the anther develops, the cell of the sporogenous tissue will undergo meiotic division. Okay, to form microspore tetrads. Okay, what is a tetrad? Four cells together, we call it as a tetrad. Okay, so as each cell of the sporogenous tissue develops, tissues. Sorry, okay. I'll start again, okay? So microsporogenesis means the generation of microspores. So as the anther develops, the cells of the sporogenous tissues will undergo meiotic division. So this will form a microspore tetrad. Okay, so each cell in the sporogenous tissue will be capable of producing a microspore tetrad. Clear? So each of this tetrad will be capable of producing a potential pollen or the microspore mother cell. Okay, so this process of formation of microspores from a pollen mother cell through meiosis is called as microsporogenesis. Clear? So the microspores, as they are formed, they are arranged in cluster of four. As I already told you, tetrads is another cluster of four. Okay, as the anther matures, what will happen? It will dehydrate. All the water in the anther will get what to say, drained. Okay, so it will shrink, and the microspore will dissociate from each other and they develop into a pollen grain. Okay, so you can see here in the final layer the another the sporogenous tissue here. Okay, so once the microsporogenesis occurs, in the anther it will mature, it will dehydrate ஆனா என்னாகும், water எல்லாம் போய் it will shrink. Okay, and the microspore here at the microspore here, it will dissociate and it will form dissociate from each other and it will form a separate pollen grains. So these are the separate pollen grains. This is the diagram of a dehisced anther. Okay, so inside each of the microsporangium, several thousands of microspores or pollen grains will be formed. Okay, which will be released with it from the dehisced anther. Okay, so this is the microsporogenesis process. Clear? Okay. So microsporogenesis na enna we saw it is a formation and differentiation of microspores. Okay. So for initially the sporogenous tissue and the sporogenous cells form ஆகும். Each sporogenous cells is capable of producing a microspore tetrad. This microspore tetrad, what will happen? It will give rise to uh, each microspore tetrad will be capable of producing a pollen grain. And finally, when the anthem matures, it will get dehisced and separate individual pollen grains uh, will be released. Okay. Okay. Now we'll see about the pollen grain. What are its structures? What are its layers? Everything. Okay. So what is a pollen grain? How will it be? It will be generally spherical in shape. Okay, round-ஆ இருக்கும், spherical-ஆ இருக்கும். என்ன measurement-ல இருக்கும்னா generally 25 to 50 micrometers தான் இருக்கும் in diameter. Okay, and it will have a prominent two-layered wall. ரெண்டு layered wall இருக்கும். So the outer layer, the hard outer layer வந்து exine. Okay, so whatever is outside, external னு சொல்லுவோம். So X னு சொல்லுவோம். So exine. Okay, so this exine is made up of sporopollenin. Clear? So anga nama paathu tapetumla we saw about sporopollenin. Okay, so this outer layer called exine will only be made up of sporopollenin. So what is the sporopollenin? It is the most resistant organic matter ever known. Okay, so it is very resistant. Clear? Okay, so this sporopollenin, it is very resistant. It is the most resistant organic matter known. It can withstand high temperature, strong acids, alkali, and no enzyme. Uh, and no enzyme that degrades sporopollenin is so far known. Clear? Okay, so the pollen grain exine has a prominent aperture called as germ pores. Okay. Mm, okay. So this is the exine structure here, and this is the intine structure. Okay. So we were seeing about the pollen grains. Exine has a prominent aperture called as germ pores. Okay, where the sporopollenin is absent. Clear? So this sporopollenin will be absent in the germ pores. You can see some pores here. Okay, openings mari inga vandu irukku. So in this the sporopollenin will not be there. So this is the germ pore structure. Clear? And the pollen grains are well preserved as fossils because of the presence of sporopollenin. Clear? And the exine exhibits an array of patterns and designs. So the exine will be in various patterns and designs. So the outermost is the exine, the inner structure is the intine. Clear? So the inner wall of the pollen grain is called the intine. So it is a thin and continuous layer made up of cellulose and pectin. We already saw it is made up of cellulose, pectin, and the cytoplasm of the pollen grain is surrounded by a plasma membrane. Okay, so pollen grain என்னோட structure எப்படி இருக்கும்? It will be spherical, diameter வந்து 25 to 50 micrometers in diameter வரைக்கும் இருக்கும். It has a two-layered wall. One is an outer layer is called as exine, and the inner layer is called as intine. Okay, so exine is made up of a structure called is made up of sporopollenin. It is a material which is the most resistant. Okay, it can withstand high temperature. Resistant na, it can withstand many extreme conditions like extreme temperature, extreme acids, very strong acids, alkali, எல்லாத்தையுமே வந்து it can withstand. Okay, then there is a structure called as germ pores. சில structures இருக்கும் where this in the exine the sporopollen will not be there. That is known as germ spores. Okay, then likewise the exine will be of various patterns and designs. Then the inner wall is called as the intine. Inner wall, so intine, it has a thin continuous layer made up of cellulose and pectin. Okay, and then we have the cytoplasm of the pollen grain, which is surrounded by the plasma membrane. Okay, so when this pollen grain matures, what will happen? It contains a two cell. Maturation nadandichuna, it will have two cells, the vegetative cell and the generative cells. Rendu cells irukku, vegetative cells and the generative cells. So vegetative cell eppadi irukum? It will be bigger. It will have abundant food reserve. Nariya food reserve irukum, periya size la irukum, irregular ah irukum. Okay, irregularly shaped nucleus irukum. So this is the vegetative cell. It is bigger, it has abundant food reserves, and it is large irregular shaped nucleus. Clear? Then is the generative cells. So what is a generative cell? It is a small, it floats in the cytoplasm of the vegetative cell. So this is the generative cell. Here வந்து smaller the cytoplasm la வந்து the vegetative cell oda cytoplasm la float ஆயிட்டு இருக்கும். That is known as generative cells. So once the pollen grain matures, ரெண்டு type of cells வந்து form ஆகுது, vegetative and generative. Clear? So in 60% of angiosperms, 60% of the flowering plants, two cell stage நடக்கும். The pollen grains will be shed at the two cell stage. ரெண்டு cell இருக்கிற இந்த stageல வந்து it will shed. Okay, and in the remaining species, in the remaining 40% of the species, what will happen? The generative cell will again divide mitotically to give rise to two male gametes. Okay, so most of the angiosperms, அதாவது 60% of the angiosperms என்ன ஆகும்? இந்த pollen grains வந்து இந்த two cell stageலயே வந்து shed ஆயிடும். But in the remaining 40% ல என்ன ஆகும்னா, இந்த generative cell will again mitotically divide to form two male gametes. Okay, and this two male gametes will then shed the pollen grains. Okay, this is known as a three cell stage. Okay, so another two types of stages, two cell stage and three cell stage. 60% of angiosperms two cell stage lay when the pollen grains will shed, whereas in the other remaining ones only in the three cell stage it will shed. Okay, so this is about the pollen grain. So these are the pollen structures, pollen grain or the structure we already saw in the exine, intine. Okay, இதெல்லாம் பார்த்தோம். Once it matures, it will form the vegetative and generative cells. Clear? Okay. Now, apart from reproduction, apart from in reproduction, what are the other harms of pollen grains? Okay, you would have heard about pollen allergies. So these pollen grains have capacity to cause severe allergies. It will cause bronchial affliction. So what is affliction? It is causing a harm or pain in the bronchus. Okay, that is bronchial afflictions. Okay, it is also capable of producing chronic respiratory disorders. Okay, it is capable of producing chronic respiratory disorders like asthma, bronchitis, etc. Okay, so when wheat was exported from other countries to India, what happened? There was this Parthenium plant and carrot grass. They were contaminated. The wheat which was exported was contaminated with Parthenium or carrot grass. Okay, so this had those pollen grains which caused affliction, asthma, bronchitis, and pollen allergies in many people. Okay, so this is one example of pollen allergy. Okay, So we saw what are the harms it can cause. So what are the benefits of pollen grains? So pollen grains, they are rich in nutrients. Okay, pollen tablets are used as food supplements are use பண்றாங்க. Tablets for syrups அது மாதிரிலாம் யூஸ் பண்றோம். Okay, and also it is given for increasing the performance of athletes, race horses, etc. Okay, now what is the viability of pollen grain? Okay, viability na enna? How long it can live? Okay, அதுதான் how long it can viable how long it can live, அதுதான் வந்து viability. So viability depends upon the temperature and humidity. It will depend upon temperature and humidity. Clear? So சில plants ல பார்த்தோம்னா it will be only for few minutes. The pollen grains, it can survive only for few minutes. For example, in rice, wheat எல்லாம் only few minutes it can survive. Whereas the Solanaceae, Rosaceae in the family எல்லாம் வந்து it can survive for few months also. Okay, so the viability varies from plant to plant. So for this reason, what happens there, they have created something called as a pollen bank. Okay, pollen bank na enna? These pollen grains of large number of species will be stored for years in liquid nitrogen. Okay, in the liquid nitrogen la vandu minus 196 degrees Celsius they store it. Okay, this process of storing in liquid nitrogen is known as cryopreservation. Okay, so liquid nitrogen radnala the term வந்து cryo we call it as. Okay, so cryopreservation use பண்ணி இந்த method use பண்ணி the pollen grains are stored even for years. Okay, so till now what all we saw? We saw about the we saw what is sexual reproduction. We saw what are the aid of flowers, what are the aid of sexual reproduction, what is the end product of sexual reproduction. அப்புறம் 3 stages இருக்குன்னு பார்த்தோம். அதுல first வந்து pre fertilization stage we have started. Okay, so இதுல வந்து we have started the parts of the flower. Okay, so accessory parts பார்த்தோம் and the reproductive parts பார்த்தோம். So அதுல we have seen the stamens alone. Stamens are the male reproductive part. So stamens வரைக்கும் we have seen. Till here someone has any doubt? Okay, இது வரைக்கும் ஏதாவது doubts இருக்கா? If you have any doubts, you can stop me here. Any doubts? Others? Okay, Rohini has responded. Others? Okay. Others, what happened? Okay, so we'll move on to the next part. So we saw about the male reproductive organ. Now we'll see about the female reproductive organ. Okay, which is the pistil. Okay, so the pistil or the carpel. So this is the female reproductive organ. So அங்க நம்ம என்ன சொன்னோம்? The male reproductive structure is called as the andrecium. So here the female reproductive structure is called as the gynecium. Clear? So gynecium is composed of 3 parts. The female reproductive part is composed of 3 parts: stigma, style, ovary. Okay, so stigma, style, and ovary. Okay, the swollen tips of the stigma is nothing but the swollen tips of the pistil. Clear? So what is the main function of stigma? It will be covered by sticky substances for pollen grains to adhere. Okay, so in the stigma என்ன இருக்கும்? Sticky substances இருக்கும். So this pollen grains, when they come and deposit, it will get adhered to it. Okay, that is the main function. Okay, and they are specialized for receiving the pollen grains. So they are specialized for receiving pollen grains. So what is the function of next is the style. Stigma patro, next is your style. The long slender tube that connects the stigma to the ovary is called as the style. Okay, so what is it? Function என்னது? It acts as a conveyor. Conveyor na or carrier mari. ஒரு பிளேஸ்ல இருந்து இன்னொரு பிளேஸ்க்கு pollen grains transport பண்ற job. Okay, will be done by your style. It is the long central slender tube that connects the stigma to the ovary. Okay, so the third part is your ovary. So what is the ovary? It is the enlarged basal portion of the pistil. The enlarged basal portion of the pistil is called as your ovary. The ovary houses the ovule. Ovary kulla da vandu ovules irukum. Ovules da vandu it contains the egg cells. Okay, it contains the eggs. Okay, so இப்போ இப்படி ஓவரிய வந்து cross check பண்ணும்போது what are the different structures seen? Carpel, locule, ovule, funiculus and placenta. So these are the various structures seen when the ovary is cross sected. Okay, we'll see each one of them. Okay, first is the carpel. So the segment or the partition that comprises an ovary is called as the carpel. So this is the carpel. Okay, so the ovary wall supports the ovary. So inga veliya irukkiradhu vandu ovary wall. This will support the ovary, and the segment or the partition that comprises an ovary is the carpel. So this ovary, it has 3 carpels. Okay, next is your locule. Okay, what is the locule? It is the space or the cavity within the carpel. So inga irukra space, or space or cavity or gap irukla, so that is the locule. Okay, and ovary with 3 carpel na adukku 3 locules da irukum. Okay, how much of a carpel it has, based on that it will have the locule. Okay, okay, now if when a plant ovary is cross sected, you can see this part is the carpel, inside this cavity is the locule, and these are the ovules. Okay, so here there are 2 ovules per locule. Okay, rendu ovule irukku, oru locule la rendu ovule irukku, and together this part is your carpel. Okay, clear? Okay, so then there is a structure called as ovary core and placenta. So the core nale enna, the middlemost, the innermost. So this is the ovary core. So this part is the ovary core. Okay, it is the solid structure at the center. Okay, and the edge of the core is called as the placenta. Edge of the core is the the edge of the core is the placenta. This will nourish the egg. Clear? This will nourish the egg. So you can see here. So for visual representation, you can see here the core na the middle la irukadhu core, and the sutti irukadhu the placenta. Okay, so here it is the core and it is the placenta here. Here also you can see. Clear? Then a structure called as funiculus. So what is a funiculus? It is a short stack that connects the ovule and the placenta. Okay, so idana ovule, the ovule placenta oda connect பண்ற stack. இதுதான் வந்து funiculus. Sorry. So this stack is called as the funiculus. Okay, then you have the ovules. Ovules da vandu produces the egg. So these are the ovules. Clear? Okay, so enna structures paathom? Carpel, carpel, locule, ovule, funiculus, placenta. Okay, ellame paathachu. These are the various structures. Now we'll see the types in carpel. So what are the various carpels? There are 2 main types. It can be either monocarpic or multicarpellary. Multicarpic. So mono means 1. So monocarpic na single carpel. Okay. Multicarpellary na more than 1 carpel is known as multicarpellary. Clear? Okay. So the gynoecium may consist of a single pistil or carpel, which is monocarpellary, or it can have more than 1, that is multicarpellary. Okay, so when there are more than 1, the pistil may be either fused or it can be free. Okay, so multicarpellary ah irukumbodhu, more than 1 ah irukumbodhu enna agum, and the pistil vandu fused ah irukalam. joined fused means, or it can be free, taniya irkalam. So fused and that is called as syncarpus, and if it is free, it is called as apocarpus. Clear? So you can see here, okay, so this is fused, so this is syncarpus. Okay, here it is separate, it is free. Here the carpels are free, so it is apocarpus. These are the types of carpel. Okay, next we'll see about the structure of the ovule, the megasporangium. Ovule is the megasporangium. Okay, so இதுதான் வந்து ஓவரியோட structure. So it has the following parts. Okay, it has an integument, nucleus, funiculus, embryo sac, a micropyle, and a chalaza. These are the various parts of a ovule. Okay, and the ovule பார்த்தோம்ல, அந்த ovule குள்ள வந்து இவ்ளோ parts இருக்கு. Okay, you can see the integument. Okay, here this is the integument, the outer and the inner integument. Okay, so outer and the inner integument, they are nothing but protective coat layers. Protective layers, வெளியில இருக்கிற protective layers தான் வந்து integument. Okay, so skin வந்து என்ன சொல்லுவோம், integumentary organ னு சொல்லுவோம். So வெளியில இருக்கிற protective layer. Okay, அதனால தான் here also these two, outer and inner integuments are protective coats for the ovule. Okay, next is your nucleus. Nucleus, where it is a mass of tissues consisting of meristematic. Meristematic is dividing cells. So it is a mass of tissue which consists of the meristematic cells. Okay, that is present here. This is the nucleus here, nucleus. Okay, and then we have the funiculus. Okay, what is a funiculus? Here you can see the funiculus. Funiculus is the part which attaches the ovule to the placenta. The ovule placenta kuda attach pandra part we already started. The small stalk which attaches the ovule to the placenta, adudha vandu funiculus. Then we have the embryo sac. Embryo sac inga irukku. So this is the embryo sac which contains the eggs. Okay, then we have a micropyle. Okay, here it is the micropyle, a small opening at the tip here. It is called as the micropyle, and the basal part of the ovule is called as the chalazal pole. Okay, so this is the basal part. கீழ இருக்கிற part, the base part, the basal part is chalazal. Clear? So these are the various structures of an ovule. Okay, the integument, nucleus, funiculus, embryo sac. So these are the various structures of the ovule. Now we'll see the types of ovule. என்னென்ன மாதிரி types இருக்கு. Okay, there is various types, 6 types இருக்கு. Orthotrophous. Orthotrophous na the micropyle, chalaza, funicle are in the straight line. Straight line ah irundhuchna it is orthotrophous. Okay, so the ovule turns at 180 degree angle and inverted ovule. So 180 degree angle turn பண்ண என்ன ஆகும், inverted ஆயிடும் ovule. அந்த மாதிரி இருக்கிறத வந்து we call it as anatrophous. Okay, and then there is something called as camphylotrophous. Camphylotrophous na the ovule will be curved more to the right angle of the funicle. அப்படி இருந்தா we call it as camphylotrophous. Okay, then hemiantrotrophous. Okay, hemiantrotrophous means ovule will turn 90 degree angle upon the funicle or the body of the ovule. Okay, right angle to the funicle irundhuchna it is hemiantrophous. And amphitrophous na enna, the ovule will be as well as the embryo sac will be curved like a horseshoe mari irukum, this diagram. Okay, so horseshoe madri irundhuchna it is amphitrophous. Okay, then we have circinotrophous. That means the ovule will turn at more than 360 degree angle. So more than 360 degree angle full turn eduthuchna it is circinotrophous. Okay, for each of them there is various examples. For example, piper, the pepper producing plant, that is orthotrophous example. And for each of them we have various examples. So this is about the different types of ovules. Clear? Okay. Okay, now we'll see the development of the gametophytes. Okay, the two stages, the megasporogenesis and the megagametogenesis. Okay, what is megasporogenesis? Genesis nale generation, formation. Okay, so megasporogenesis na formation of megaspores and the megasporogenesis. So methanogenesis na, it is the creation, the formation of gametes. The egg form பண்றதா வந்து megagametogenesis. Here we'll see about these two. Okay, first we'll see about the development of the gametophytes. That was the megasporogenesis. Okay, so mega nale it denotes to the egg. Okay, micro means to the male reproductive structure, the pollen grains. Okay, so here the megasporogenesis enna aguna, the ovules will produce megaspore mother cells, which is diploid 2n. Okay, this will undergo meiosis. Okay, the megaspore mother cell will produce 4 megaspores via 2 times meiosis. 2 times meiosis will occur. Okay, 2 times meiosis will occur and it will form 4 megaspores. Tetrads form pannum. Okay, and 3 of the megaspores will die and only 1 will be left to continue. Okay, so this is the process. Megaspore enna aguna, ovule will produce megaspores, and this megaspore mother cell will produce 4 megaspores via 2 meiosis. 2 times meiosis will occur and 4 megaspores will be formed, and 3 of these megaspores will die and only 1 will be left. Clear? Okay, next is megagametogenesis. Gamete form பண்றது, development of gametophytes. Okay, so இதுல என்ன ஆகும்னா, the surviving megaspores will divide via mitosis 3 times. Okay, forming 8 haploid cells. Okay, so mitosis 3 times நடக்கும். Okay, 1 2 3. So 3 times in the megaspores will undergo mitosis and they will form 8 haploid cells. 8 haploid cells form பண்ணும். So this is megagametogenesis. Clear? Okay, so how will the egg develop in the ovule? Okay, so among the 8 cells we saw, among the 8 cells, 3 cells will migrate near the micropyle. Micropyle na enna, and the tip la irukra opening. Okay, the tip la irukra opening is known as micropyle. So idhula vandu 8 cells la enna agum, 3 cell vandu it will go near the micropyle. Okay, and the one at the center will become the egg, while the other 2 will become the synergid cells. Okay, so ipo vandu 8 cells la vandu 3 cells vandu micropyle kitta poidum. Okay, one vandu center la irukum, apra other 2 vandu then other 2 will go to the top. Okay, 3 4 5 6. Okay, so the remaining 2 I will see later. Okay, so now 3 cells will migrate to the micropyle, 1 will be in the center, 2 will become synergid cells. Clear? So the other 3 migrates on the opposite pole and they will become antipodal cells. Remaining 2 cells will be naked, there will be no membrane, they will remain at the center to become the polar nuclei. Okay, so this is the antipodal, this is the polar nuclei, and these are the synergids. Okay, these 8 cells will form the inside of the embryo sac and are ready to be fertilized by the pollen from the male. Okay, so these 8 cells, idoda structure வந்து இந்த மாதிரி ஆயிடும். Here there will be antipodals, நடுவுல வந்து polar nuclei, இதெல்லாம் synergids, here will be the egg cells. Okay, so 3 4 5 6 7 and sorry, so these are the 8 cells from the inside of the embryo sac and are ready to be fertilized by the pollen from the male. Okay, so the embryo sac development, how will the embryo sac develop? So initially pathona, the ovule will be developing. Here is the developing ovule. Okay, so this developing ovule, the position of the megaspore mother cell will be buried in the nucleus tissue. Here will be the nucleus tissue. Okay, it will the diploid megaspore mother cell will undergo meiosis Okay, and it will form the megaspore. Okay, and the developing embryo sac uh, and will develop the 2 cell and the 4 and the 8 form பண்ணிடும். Okay, and it will become a mature embryo sac. So the in the developing ovule இருக்கிற placeல the mature embryo sac will be present. Okay, this is the embryo sac development. Okay, okay, so this is about the uh, so now we have seen about the female reproductive structures, ovules, what are the different parts, what are the types of carpels, what are the structure of an ovule, then we have we saw the types of ovules, then we saw the development of gametophytes. We saw megasporogenesis, then we saw megagametogenesis, then we saw the egg development in the ovule, then the embryo sac development in the ovule. Okay, clear till here? Clear till here? Okay. So next is pollination. Okay. So pollination. So what is pollination? Transfer of pollens from anther to the stigma is called as pollination. So anther lende pollen grain stigma ku transfer agra process is known as pollination. Okay. So pollination can either be self or cross. Okay. Self pollination na transfer of pollens from an anther to stigma of the same plant. Whereas two different plant of the same species nadandichuna, it is cross pollination. Clear? So these are the two types of pollination, self and cross. Okay, so what are the various categories? அதுக்கு கீழ என்னென்ன categories இருக்கு பார்க்கலாம். So self pollination, now what I told you, it is a pollination which occurs in the same flower but different flower, I mean same flower or the different flower of the same plant is known as self pollination. So self pollination is of two types, can be autogamy or geitonogamy. Okay, so autogamy means pollination in the same flower, geitonogamy means different flowers of the same plant. Okay, so autogamy can be further divided into chasmogamy and cleistogamy. Okay, clear? So chasmogamy means the flowers will open, the synchrony will it will synchrony in the pollen releases and stigma. Sorry, here it is not clear. We will see it in detail later. Okay, so chasmogamy and cleistogamy. So cleistogamy is flowers which do not open at all, whereas chasmogamy there'll be a synchronization with the pollens and it will open. Okay, so that is the main difference here. Okay, next we saw cross pollination. What is cross pollination? Two flowers of different plants of the same species will pollinate. That is known as cross pollination. Okay, so here we can see xenogamy. What is xenogamy? Transfer of pollen grains uh, from different plants. Okay, so this is the only method of pollination which will bring different types of pollens during pollination. Okay, whereas in other types only one from a single flower or from a single type the pollens will be brought. Whereas here different types of pollens will be brought during pollination. Okay, so pollination is of two types, self pollination, cross pollination. Self la வந்து we have autogamy, geitonogamy, and in autogamy we can see chasmogamy and cleistogamy. Okay, cross pollination we have seen xenogamy. Clear? Okay. So next is agents of pollination. So in the who who is bringing about pollination? எந்த மூலியமா pollination நடக்குது? Okay, so pollination can occur either by abiotic methods or biotic method. Okay, abiotic na through wind, water, அதெல்லாம் வந்து we call it as abiotic, whereas biotic is through any living organisms like insects, birds, bats, snail, or any other larger animal, even including humans. It is biotic. Clear? Okay, so biotic pollination. So biotic pollination, 80% of pollination is biotic, that is through by insects. Okay, insects nala pollination நடக்கிறதுக்கு பேர் என்ன? Entomophyly. Okay, insects na entomo சொல்லுவோம், so entomophyly. Okay, so if it occurs through butterflies, it is called as psychophyly. If it occurs through moths, it is phalaenophyly. Flies na sapromyophyly. Okay, if it is through birds, it is ornithophyly. If it is through bats, it is chiropterophyly. Okay, so this is how the bees will transfer from the pollens from one flower to another. Okay, next is how the insect pollination, the adaptations of animals or insect pollinations. Okay, since the petals are brightly colored and scented with nectars, small amount of sticky what will happen, these insects will approach these brightly flowered scented flowers with nectars. Okay, when they sit when they sit on these flowers, what will happen, through through its legs, some of the pollens will get stuck, and when they go to another flower and stick on the, sit on them, the pollens will get transferred. Okay, this is the basis of insect pollination. Okay, this is about biotic pollination. Next is abiotic pollination. Abiotic pollination is by non-animal factors. So what is it? By wind and by water. So pollination by wind is known as aminophyly. Through water is known as hydrophyly. Clear? So aminophyly is pollination by wind, which is 98% of the abiotic pollination. 98% of abiotic pollination is through wind. Clear? So hydrophyly can also occur sometimes in aquatic plants. Okay, for example, hepihydrophyly, that is mediated by surface water. Surface water liye vandu in the transfer nadந்துச்சுனா. Okay, for example, in Vallisneria plants. Okay, then we have hypohydrophyly at the bottom water. Okay, surface water na epihydrophyly, bottom water na hypohydrophyly. Example in your zoostro plants. Okay, here you can see in Vallisneria, water surface la vandu you have the water surface la the pollination occurs, whereas in sea grasses inside the water the pollination will occur. In water hyacinth, water lily la vandu still it is in water plant, but insect pollination, wind pollination da nadakum. Okay, so it can either be on the water surface or at the bottom of the water. Okay, so these are the two methods. Okay, so in wind pollination, what will happen? Here the light non-sticky, they'll be well exposed to stamens. Okay, எந்த மாதிரி flowers எல்லாம் wind pollination நடக்கும்னா light non-sticky well exposed stamens, large often feathery stigma, single ovule in each ovary, inflorescence, and இந்த மாதிரி grasses, இந்த மாதிரி இருக்கிற பிளான்ட்ஸ்ல வந்து wind pollination நடக்கும். So the flower must be light, the plant must be light, அப்பதான் windல வந்து it will move and pollination will occur. Non-sticky இருக்கணும், அப்பதான் it can go and it will move through wind. Okay, then it should be well exposed. Okay, and வந்து larger இருக்கணும், feathery இருக்கணும் stigma. Okay, then it should have a single ovule. Okay, so these are the various some of the criteria for wind pollination to occur. Then we have water pollination. So water pollination is very rare. It is limited to only 30 genera. Okay, so it is mostly, it will occur in monocotyledons and water plants usually. Okay, and the distribution is limited because of the need for water for transport of male gametes. Okay, So since they need water for transport, mostly வந்து இந்த water surface ல இருக்கிற plants மட்டும் தான் வந்து இது நடக்கும். Okay, so in these plants it will have a long ribbon like pollens. Long ribbon like pollens will be there, and the pollen grains will have mucilaginous covering. It will not be colorful, and it will not have nectar also. Okay, இந்த மாதிரி இருக்கிற plants தான் வந்து water pollination will occur, especially in the water plants. Next is animal pollination. We already saw about biotic pollination. Here in specific, we'll see about animal pollination. So based on the color, fragrance, odor, okay, nectar இருக்கிற மாதிரி plants தான் வந்து flowers தான் வந்து they prefer, the animals will prefer. Okay, example is your Amorphophallus. Okay, this is the largest flower. Okay, largest flower in the world. This flower la வந்து what happens, many animals they come, they lay eggs, adukala வந்து they use. Okay, they use it for safe place, they use it as a safe place for laying eggs, and the meanwhile வந்து pollination நடக்கும். Okay, that is one thing. Another example is your moth and yucca relationship. So in central US what happens, soapweed yucca called as Yucca cloaca. Okay, this is pollinated by a moth called known as Testicular Eucacella. Okay, moth and the plant, what happens, it cannot, both the moth and the plant, they cannot complete their life cycle without each other. Okay, they're mutually beneficial for each other. The moth will deposit its egg in the locule of the ovary and the flower, and in turn they will get pollinated by the moth. Okay, so the larva of the moth will come out of the flower as the seed will start developing. You can see here. Okay, so this is the relationship between moth and the yucca. This is an example of animal pollination. Okay, so now we saw about pollination. Pollination la எத்தனை types இருக்கு அதெல்லாம் பார்த்தோம். Any doubts you have? Any doubts? Pollination we saw. What are the types? அது கீழ என்னென்ன types இருக்கு? So what are the agents of pollination பார்த்தோம். Okay, any doubts you have? Okay, so the next topic is outbreeding devices. So what is outbreeding? Okay, so outbreeding na enna, inbreeding na enna. So what is inbreeding? Inbreeding means breeding among closely related species. Okay, for example, when we are breeding a single plant, adu kitta irundhu we get up breed na, again we are breeding that plant with the parent itself. Okay, so in the very closely related species or plants la vandu if we are breeding again and again, it is known as inbreeding. Okay, so we have something called as inbreeding depression. So what is inbreeding depression? Inbreeding depression na, after some time இந்த மாதிரி inbreeding closely related இருக்கிற plants when they are breeding again and again, what will happen, the rate of survival or rate of fitness of these plants will reduce drastically. Okay, அவ்வளவா ஃபிட்டா இருக்காது plant. Okay, the organism's ability to survive and perpetuate the genetic material will also reduce drastically. Okay, அதோட it won't be a fit breed at all. Okay, அதுக்காக to overcome this inbreeding depression, there is something called as they do something called as outbreeding devices. The outbreeding devices are methods to prevent inbreeding depression. Clear? So first is pollen release and stigma receptivity not synchronized. Okay, what is it? Okay, receptivity not synchronized na, pollens வந்து release ஆகும்போது stigma should not be in a position to receive it. Okay, so அதுக்கு என்ன பண்ணுவாங்கன்னா, the pollens will be received before the stigma becomes mature, before the stigma becomes receptive, or in otherwise the stigma will become the receptive much before the release of the pollens. Okay, இந்த மாதிரி we can receptivity synchronize பண்ணாம விடலாம். Okay, we can make the receptivity not synchron, we are not synchronizing the receptivity. அது வந்து that is one outbreeding device. Next is anther and stigma will be present at different positions in the flower. அப்ப வந்து ஒரே இதுல inbreeding நடக்காது. Okay, then we have self incompatibility. Okay, what is self incompatibility? ஒரே flowerல இருக்கிற anther stigma வந்து it won't be compatible. அது வந்து it cannot what to say breed. அதுதான் வந்து incompatibility. Okay, next is production of unisexual flowers. ஒரே செக்ஸ் இருக்கிற flower produce பண்ணோம்னா this problem of inbreeding will not be there. Okay, likewise similarly we have monoecious dioecious conditions. Okay, so monoecious na enna, both male and female flowers will be present in the same plant. Okay, dioecious na vandu the stamen will be present in one flower, the pistil will be present in another flower. Okay, so these are the various outbreeding devices. We'll see about them one by one now. Okay, so the following types of devices promote and ensure outbreeding through cross pollination. Okay, first is unisexuality. Okay, so flowers வந்து unisexual we can make it. So என்ன ஆகும்னா plants வந்து either monoecious இருக்கும், அதாவது ஒரே இது ஒரே ஒரு what to say, both male and female flowers will be present in the same plant itself, or it can be dioecious, like in papaya. In papaya we have a male plant will be separate, female plant will be separate. So the male plant will produce male flowers, female plant female flowers. So இந்த மாதிரி unisexual இருந்தா ஒரு flower வந்து having only one sex இருந்தா outbreeding வந்து we can promote and we can stop inbreeding. Okay, next is dichogamy. Okay, di means two, gamo means marriage. Okay, so dichogamy means here in bisexual flower what will happen, the anther and the stigma will mature at different times. Okay, different times, so that self pollination will be inhibited. Okay, so anther mature ஆகி ரொம்ப நாள் கழிச்சு stigma mature ஆனாலோ இல்ல stigma mature ஆகி ரொம்ப நாள் முன்னாடியே anther mature ஆனாலோ what will happen, there won't be compatibility. அதனால வந்து self pollination நடக்காது. Example, sunflower la, rose la, magnolia, in all these things magnolia and all these things, what happens, dichogamy will occur. Okay, so first vandu if male vandu mature aachna it is protandry, and if the female matures it is protangyny. Okay, protandry and protogyny. Okay, so these are the two maturation things. Okay, next is your heterostyly. Okay, what is heterostyly? Flowers have different types of height of the styles and stamens. Okay, so stamen or height la, style or height and many difference, the height la difference irukumbodhu what will happen, inbreeding will be prevented. Example is jasmine, okay, prime rose, idella vandu indha madri heterostyly irukum. Okay, there will be difference in height, so we can prevent the inbreeding. Okay, next is self sterility. Okay, self sterility or incompatibility. Okay, pollen grains of one flower are incapable of growing over the stigma of the same flower due to the presence of sterile genes. Okay, sterile genes adile vandu self sterility, sterile genes vandu irukkiradhanaala inbreeding nadakradhukku chance illa. Okay, example is potato, tobacco. Okay, all these things are examples. Okay. Next is hercogamy. Okay, so these hercogamies are mechanical devices. நம்ம வந்து மெக்கானிக்கலா அத வந்து நம்ம பண்றோம். So it is known as mechanical device which prevents self pollination and favors cross pollination. Okay, so even in homogamous flowers like hibiscus, what they do, they mechanically prevent the self pollination. அது எப்படின்னா by prepotency. Pollen grains of one flower grow so mechanically we are removing one of the structure. Okay, அந்த மாதிரி இருக்கும்போது what will happen, ஒரே flowerல homogeneous flowerல கூட வந்து self pollination நடக்காது. So that is hercogamy. Next is prepotency. So what is prepotency? pollen grain of one flower will grow more rapidly over the stigma of the another flower compared to the same flower. Okay, so இதுக்கு எக்ஸாம்பிள் வந்து பார்த்தோம் ஆப்பிள் கிரேப்ஸ்ல எல்லாம் இந்த மாதிரி நடக்கும். So ஒரு flowerல இருக்கிற pollen grain வந்து it will grow more rapidly over the stigma of another flower compared to the same flower in same flower. So this is prepotency. Okay, next is artificial hybridization. So artificial we humans how we manipulate this inbreeding. So first is a procedure called as emasculation. Emasculation na enna, the female parent will bear the bisexual flower. Okay, so that bisexual flower lende we'll remove the anther from the flower bud before the anther dehisces using a pair of forceps. Or a pair of forceps vachu we will remove the anther before it dehisces. Okay, that process is known as emasculation. Then there is a process called as bagging. Bagging na enna, in the emasculate panna flower வந்து we have to cover with a suitable bag or a butter paper. அந்த மாதிரி எதுக்காக கவர் பண்றாங்கன்னா to prevent contamination from தேவையில்லாத வேற போலன்னால pollinate ஆயிடக்கூடாதுன்னு we have to cover it. Okay, this is known as bagging. Okay, so when the stigma of the bagged flower attains receptivity, mature pollen grains will be collected from the anther of the male plant and will be dusted on the stigma. So artificially what they do, once the receptivity obtain ஆனோன அந்த receptivity கான maturity obtain ஆனோன artificially we will collect the anthers of the male parent and we will dust it on the stigma. Okay, stigma dust பண்ணிட்டு என்ன பண்ணுவோம், again to prevent contamination rebag will be done. Okay, it will be again rebagged. Okay, covered. Okay, then and the fruits will be allowed to develop. Okay, so this is artificial hybridization. Emasculation nadakanu, then bagging will occur, then mature anana receptivity attain anona it will be dusted, or it will be or wantedly or artificially we will dust it and it will again be rebagged. Okay, this is artificial hybridization. Okay, unisexual flowers, no emasculation will be done. The female flower bud will be bagged before the flower opens. Okay, when the stigma becomes receptive, pollination will be carried out with the desired pollen and the flower will be rebagged. Okay, unisexual flower la வந்து இந்த emasculation process மட்டும் skip பண்ணிட்டு other things will be similar. Clear? Okay, so emasculation na enna, removal of anthers from the flower bud before the dehiscence of anther. Okay, till here everyone is clear? Okay, next is pollen pistil interaction. Okay, so this is very important, the pollen and the pistil interaction. Okay, so there will be a continuous interaction between pollen grain and the pistil. Okay, this is mediated by chemical components of the pollen and the pistil. Okay, in the pollen grainலயும் pistilலயும் ஒரு continuous interaction நடந்துட்டே இருக்கும். ஒரு wrong type interaction நடந்தா incompatible இருக்கும், whereas the suitable type interaction நடந்தா it will become compatible. Okay, so the compatible pollen grains will germinate on the stigma pollen tube through the germ tube. Okay, compatible இருக்கும்போது என்ன ஆகும், இந்த continuous interactionல compatible இருக்கிற pollen grains மட்டும் germinate ஆகும் stigmaல. Clear? Okay, so germination formation of pollen tube through the germ pore will occur, and through the germ pore the contents will move to the pollen tube through the stigma, style, and finally it will reach the ovary. Okay, so pollen grains வந்து இந்த two celled இருக்கும்போது இந்த two celled conditionல the generative cell will divide and forms the two male gametes during the growth of the pollen tube in the stigma. Whereas three-celled condition இருக்கும்போது என்ன ஆகும்னா the pollen tube will carry the two male gametes from the beginning itself. Okay, so this is the difference between two-cell generation and three-cell generation. Finally, the pollen tube after reaching the ovary will enter the ovule through the micropyle. The micropyle is the opening, so it will enter through the micropyle, then it will enter to the synergist, one of the synergids, through the filiform apparatus. Okay, so for a superior breeding, so as for superior plant development and successful breeding, this interaction, pole and pistil interaction, is very much important. Clear? Okay. Okay. So with this, we have completed the pre-fertilization stage in the sexual reproduction of flowering plants. Okay, clear? Any doubts still here? Any doubts still here? Okay. We only have 5 more minutes, so I'm stopping the class with this. Okay, we'll stop the class with the pre-fertilization stage. If you have any doubts, you can ask me now, or we'll have a short uh, test of whatever you have learned today. Self-sterility. Okay, you want me to explain self-sterility? So what is self-sterility? Okay, making the plant sterile by itself. அதான் வந்து self sterility. அதுலயே வந்து breeding நடக்காம பாத்துக்கிறது தான் வந்து self sterility. Okay, so making it sterile. So the pollen grains of one flower are capable of growing over the stigma of the same flower due to the presence of similar sterile genes. Okay, so ஒரு சேம் ஃபிளார்ல இருக்கிற போலன் கிரெயின் வந்து அதுலயே இருக்கிற ஸ்டிக்மால க்ரோ ஆகிறதுக்கு, ஐ மீன் பாலினேட் ஆகிறதுக்கு வந்து will not occur. It will be incapable of growing over the same stigma of the same flower. This is because of sterile genes. Okay, so artificially what they do, they will inject some sterile genes in it. Okay, so this sterile genes, what it will do, it will not allow the same plants to same plants to pollinate. I mean, ஒரே plantல இருக்க flowers to pollinate. Okay, so that is known as self sterility. You're clear? Should I explain again? Yes, Kavya, you have raised the hand. You want me to unmute you, or you have any doubts? Please type in the chat box. What doubts you have? Okay, so this is because what is a megaspore? A megaspore means a egg. Okay, so egg will be, egg வந்து எப்பயுமே ஒன்னு தான் வந்து it will produce. Okay, we are clear. megaspores வந்து ஒன்னு தான் இருக்கும், but microspores will be many. Okay, so this is the basic trivia or the basic concept. Egg will be one, whereas the male reproductive structures, sperm or any pollens, இதெல்லாம் வந்து நிறைய இருக்கும். So that is the basic concept. So here a single megaspore mother cell is giving rise to 4 megaspores. Okay, so 4 megaspores வச்சு we can't, I mean it is not needed for us. Okay, so we need a single fittest megaspore, we need a single egg. So only one will be left and all other 3 will die. Clear? Kavya, you're clear with this concept? Okay. Any other doubts? Sorry for the connection issues at the beginning. Okay, because of the rain. Any other doubts? Okay, it is almost time. Okay, all your test results I'll message you now after the class. Okay, your answers will also be sent to the group. Okay, I'm sorry today we didn't have much time, so next class we'll discuss about the uh, test questions. Okay, test answers. Okay, I'll anyway send it to you all. Okay. Okay, thank you all. Since there are no other doubts, I'm closing the meeting here. Thank you. Next class we'll see the fertilization and post fertilization. Thank you.